everyone, we're going to go through and talk about how to go ahead and make a microscope slide so that you can look at animal cells. And specifically, the cells that we're going to look at from animals this time are going to be human cells. So we are going to look at human animal cells underneath the microscope. And the cells that work really well for this sort of an assignment, for this sort of a task, are going to actually be cheek cells. So we are going to use human cheek cells in order to look at underneath the microscope. And so when we take these cheek cells, we're not taking them from the outside part right here. We're going to actually take them from right in here, those cheek cells in there, because it turns out you have a lot of them and they are extra and you don't always need that many, so they're really convenient. So in order to do this, we're going to start off by taking a toothpick, so just a normal toothpick, and I'm going to scrape along the inside of my cheek. So I'm going to scrape. Alright, so right now I've got thousands of Mrs. C's cheek cells hanging out on this toothpick. And what I'm going to do is I'm going to take this and I'm going to smush them. That's the technical term. I'm going to smush them onto this slide. So I'm going to just kind of spread those cheek cells around here on this cell. So there they are. They're on there, okay? But they're really tough to see yet, you guys. And they're really tough to see because cheek cells are transparent. That means you can see right through them. They don't have a lot of color to them. So because they are transparent, what we're going to do is we have to stain them. We have to color them so that we can actually see them underneath the microscope. If we looked right now, it would be really tough to see anything because, like we said, they're see-through, they're transparent. So what we're going to use is going to be something called iodine. It's a fancy sciencey looking thing. And this iodine is basically a stain. So my farm kids, you have probably used iodine before because people also use it as a disinfectant to kind of get rid of any bacteria or germs that might be living on places. So people use it on their cows a whole bunch. But we're going to use it just as a stain this time. So I'm going to go ahead, I'm going to put 1 drop of that iodine here on my slide. And just like we did with the pond water, I'm going to take a cover slip and I'm going to put that on top so that it kind of smooshes it out and makes it all nice and flat. And now I have a whole bunch of Mrs. C cheek cells here on my slide ready to go look at underneath the microscope. I went ahead and put my slide on the stage of my microscope and I went to go find some cells, and this is one kind of item that I found. Now this is actually a whole bunch of cells all stacked up on each other. So every one of these little circle blobby looking things, every single one of those is a Mrs. C cell. So for this lab, what we really want to be able to do is we want to be able to find just one cell kind of hanging out by itself so we can really see what's going on with these cells. And there's a really good one. Not to brag, you guys, but I've got a pretty phenomenal cheek cell right there. So this right here is going to be this really good cheek cell that we're going to use to help us identify some different parts and what's going on. But it's not like I just took one cheek cell out of my mouth. All of these are cheek cells, and we can see that some of them are kind of stacked up on one another.